Good morning everyone. I am Dr. Kalpana Sharma, Associate Professor, Department of Zoology, S.P.C Government College, अजमेर। बच्चों, मेरे लास्ट टर्न पर मैंने आप लोगों को क्लोनिंग पार्ट 1 बताया था, जिसमें कि हमने व्हाट इज़ क्लोनिंग एंड हाउ मेनी टाइप्स ऑफ क्लोनिंग आर देयर के बारे में डिस्कस किया। देयर वर बेसिकली 3 टाइप्स ऑफ क्लोनिंग: रिप्रोडक्टिव क्लोनिंग, जीन क्लोनिंग एंड थेरेप्यूटिक क्लोनिंग। लास्ट टर्न पे हमने जीन क्लोनिंग के बारे में डिस्कस कर लिया था। आज मैं आप लोगों को रिप्रोडक्टिव क्लोनिंग, थेरेप्यूटिक क्लोनिंग के बारे में डिस्कस करूँगी। सो इन दिस यू कैन सी द रिप्रोडक्टिव क्लोनिंग. This type of cloning produces copies of whole animals. Reproductive cloning के अंदर पूरे animal का निर्माण हो जाता है. This usually involves a method in which a mature somatic cell is removed from an animal and DNA transferred into an egg cell whose nucleus has been removed. Egg is then allowed to develop into an early stage embryo in the test tube and is then implanted into the womb of an adult female animal. Ultimately, the adult female gives birth to an animal that has the same genetic makeup as the animal who donated the somatic cell. This technique mainly uses the nuclear transfer method. In sexual reproduction, the zygote receives half of the genes from the mother and half from the father. If this embryo splits, it divides into 2 genetically identical embryos, which are then able to divide independently. This results in genetically identical twins. In reproductive cloning, the entire genetic code is reproduced from a single body cell of an adult individual. This technique used is SCNT, which is somatic cell nuclear transfer technique. Ruzo Yanagimichi et al. Of Hawaii University discovered a new technique after changing King Briggs and Godon's technique and named it as Honolulu technique. What is Honolulu technique? In this technique, the nucleus of the cell is from G0 to G1 stage are placed in the enucleated egg cell and clones are obtained. Mice has been cloned using this technique. Now next is the therapeutic cloning. Cloning designed as therapy for a disease. ऐसी cloning जिसको हम किसी therapeutic purpose के लिए use करते हैं, disease के diagnose के लिए use करते हैं. In therapeutic cloning, the nucleus of a cell, typically of a skin cell, is inserted into a fertilized egg whose nucleus has been removed. The nucleated egg begins to divide repeatedly to form a blastocyst. Scientists then extract stem cells from the blastocyst and use them to grow cells as are a perfect genetic match for the patient. The cell created via therapeutic cloning can be transplanted into the patient to treat a disease from which the patient suffers. In contrast to the goal of therapeutic cloning, the goal of reproductive cloning is to create a new individual, an idea that has stirred great controversy and met with almost uniform disapproval. This type of cloning involves harvesting of stem cell that can be used for research and therapy. The major difference between reproductive cloning and therapeutic cloning is this, that in the later, like that in the therapeutic cloning, the embryo is not implanted in the womb. Instead, stem cells are extracted. This technique opens new ventures for research and therapy. Now, in this picture, you can see the method of producing a clone of donor, in which you can see on the left side the egg cell is there, in which there is a nucleus. इस न्यूक्लियस को यहाँ से रिमूव कर रहे हैं, मतलब इन्यूक्लिएशन कर दिया. ऑन द राइट साइड में एक और सेल है जिसके अंदर न्यूक्लियस है और डीएनए है. इसका यहाँ से डीएनए एक्सट्रैक्ट कर लिया. अब ये सेल इन्यूक्लिएटेड सेल हो गई और इन्यूक्लिएटेड सेल के अंदर डीएनए जो था डोनर का वो प्लेस कर दिया. नाउ व्हेन दिस सेल विल डिवाइड, द एम्ब्रियो विल बी प्रोड्यूस्ड and the implanted in the womb of the same donor animal whose cell was used. अब इसको एम्ब्रियो को क्या करेंगे, वूम के अंदर इम्प्लांट कर देंगे. किसके अंदर, जो कि सेम डोनर एनिमल whose cell was used. बर्थ टू अ क्लोन ऑफ डोनर. अब इस बिल्कुल डोनर के जैसा क्लोन बन जाएगा. तो ये वाली जो टेक्निक है ये तो है रिप्रोडक्टिव क्लोनिंग, क्योंकि इसके अंदर जब सेल डिवाइड करती है और एम्ब्रियो जब हो जाता है तो उस एम्ब्रियो को हम वूम के अंदर ट्रांसप्लांट कर देते हैं. वेयर एज इन थेरेपेटिक क्लोनिंग वी डोंट इम्प्लांट द egg into the, we derive the stem cells from it for the therapeutic purposes. Now here you can see the cloning. Clone is done at 4 bases: molecular basis, cell basis, embryonic basis and organism basis. In molecular cloning, we clone it at gene level. Cell cloning, we clone the cells only at a cellular level. And embryonic cloning, we done it as embryonic level. And organism cloning done at plants or animal level. Or clones of the whole organisms are made. In molecular cloning, clones of DNA, chromosomes are made. In cell cloning, clones of cells are made. In embryo cloning, embryos are made. Whereas in organism cloning, whole organisms are made. Now in this, you can see the therapeutic cloning. We are doing the same procedure. That what we are doing here on the left side, you can see we are enucleating a cell, जिसका हमने न्यूक्लियस निकाल दिया और ये सेल खाली हो गया है. राइट साइड पे अनफर्टिलाइज्ड एग है, उसके अंदर न्यूक्लियस के अंदर से हमने डीएनए एक्सट्रैक्ट कर लिया है और इस डीएनए को हम उस इन्यूक्लिएटेड सेल के अंदर ट्रांसफ़र कर देते हैं. इसमें जब सेल डिवाइड होगी तो आफ्टर 4-5 डेज एम्ब्रियो का निर्माण हो जाएगा. अब इस एम्ब्रियो से हम स्टेम सेल को रिमूव कर लेंगे और इसको हम मदर के वूम के अंदर ट्रांसफर नहीं करेंगे. क्लोनिंग इन मैमल डॉली. द फर्स्ट मैमल क्लोन फ्रॉम एडल्ट स्टेम सेल बाय सेंट टेक्निक वॉज अ शीप नोन बाय द नेम डॉली. जो SCNT टेक्निक के द्वारा तैयार किया गया था वो डॉली था. SCNT मीन्स सोमैटिक सेल न्यूक्लियर ट्रांसफर टेक्निक. She was a female domestic sheep and cloned by Ian Wilmot and Campbell and co-workers at the Roslin Institute, University of Edinburgh, Scotland. She was born on 5th July 1996 and died of a progressive lung disease 5 months before her 7th birthday. The technique had the following steps. A pregnant Dorset sheep was taken and its udder cells were extracted. These cells were deep freezed and fed with a little nutrition, which made them lose their udder cells ability and their gene became less active or went in G0 or G1 state. Now, these cells were ready for any reprogramming. पहले एक प्रेग्नेंट डॉर्सेट शीप ले लिया, उसकी अडर सेल जो थी उसको हमने एक्सट्रैक्ट कर लिया, फिर उसको रीफ्रीज कर दिया. इससे क्या, वो G0 G1 फेज के अंदर चली गई. अब इन सेल को हम रीप्रोग्रामिंग के लिए यूज़ कर सकते हैं. Another Scottish sheep was taken and its egg cell were extracted and enucleated. Both the udder cells and enucleated egg cells were kept in a culture medium and electric shocks were induced. This led to the fusion of the cells, and the enucleated egg cell incorporated udder cell DNA in it. Egg cells now started dividing, and undercell DNA also got reprogrammed according to the new environment. When this dividing hybrid cell developed into a blastocyst implanted in a surrogate mother. This surrogate mother gave birth to a female sheep on July 5th, 1996, named Dolly, whose weight was 6.6 kg. Dolly sheep was a clone of a donor whose nucleus and DNA was used in the process. Dolly sheep was the first clone produced from a cell taken from an adult mammal. It also proved that the mature differentiated somatic cell also had the ability of reverting to an embryonic totipotent state. Dolly's birth was kept a secret for 6 months and announced to the public on 22nd February 1997. It was very much talked about in the media. It also raised many questions and promoted discussions on cloning and its effects on human being and the environment, but Dolly proved to be a normal sheep who gave birth to 6 lambs in total. Her first lamb was born on 13th April 1998 and was named Bonnie, whose father was a Welsh mountain ram, David. Later, in 2001, she developed arthritis, and then on 14th February in 2003, she died of a progressive lung disease and severe arthritis. Some scientists also believed that since the female Dorset sheep, whose donated udder cell DNA was 6 years in age, so Dolly also was 6 years old when she was born, and thus she died early in comparison to other Dorset sheep, whose life expectancy is usually 10 to 12 years. After Dolly, scientists demonstrated cloning in many other large mammals, including monkey, pigs, mice, and bull. 2 monkeys, Nati and Diotu, were declared to be born through cloning by nuclear embryo transfer technique. In 1997, on 12th March, in Beijing, cloned mice were produced. Cloned cows were produced by Japanese scientists. An 8-cell embryo was derived from the uterus of cow and kept in a medium where all the 8 cells were separated. Each cell was now implanted in 8 different uterus of surrogate mothers, and finally 8 clones of cows were produced. Cloned bull was also produced by American scientists. An endangered species, Bosogarus Asian gaur, was cloned by using its skin cell, which were enucleated and incorporated into an enucleated cow egg. The egg with DNA of skin cell started dividing, and after morula stage, it was transferred into a surrogate mother, which produced clones of Bosogarus donor. Scientists believed that enucleated egg contain ISWI enzyme, which can direct somatic cells to divide. Next is the human cloning. Human cloning is the creation of a genetically identical copy of a human. The term is generally used to refers to artificial human cloning, which is the reproduction of human cells and tissues. It does not refer to the natural conception and delivery of identical twins. The possibility of person cloning has raised controversies. These ethical concerns have prompted several nations to pass laws regarding human cloning and its legality. Two commonly discussed types of theoretical human cloning are therapeutic cloning and reproductive cloning. Therapeutic cloning would involve cloning cells from a human for use in medicine and transplants. That is an active area of research but is not in medical practice anywhere in the world as of July 2020. Two common methods of therapeutic cloning that are being researched are somatic cell nuclear transfer and pluripotent stem cell induction. Reproductive cloning would involve making an entire cloned human instead of just specific cells or tissues. Now we'll talk about Human Genome Project. What is the Human Genome Project, HGP? It was an international scientific research project with the goal of determining sequence of chemical basis pairs which make up human DNA and identify and mapping all the genes of a human genome. It is the world's largest biological project and started in 1984. It was proposed and funded by US. It aimed to map the nucleotide contained in a human haploid genome and sequencing euchromatic region of the genome. This project was completed in April 2003. The sequencing of human genome showcased benefit for many fields like medicine, identifying mutations, forensic medicine, protection of biogas, and agriculture, etc. It was a landmark genome project and the conclusions were termed as genomics. These conclusions and key findings were summarized in a model which explained the human genome. They are as follows. Number 1, human body has 100 trillion cells divided into 260 types. Number 2, every cell has equal number of chromosomes and thus same genome. Number 3, there are 35,000 genes in a human body and 6,000 genetic disorders. Fourth, human DNA is made up of 310 crore base pairs. Number 5, function of about 40% genes in human body are still to be explained. Now we are going to see the advantages of HGP or the Human Genomic Research Project. Number 1, the gene responsible for a particular disease can be identified and gene therapy can be implied and thus a huge gene bank can be developed. Vaccine and medicine for fatal diseases can be developed after knowing the causative gene for the disease. cDNA of all GNA will be possible. Better varieties of animals will be produced. Bioinformatics could be developed which will help in diagnosis and medication of various diseases. Human evolution will be the better understood. Forensic science will be developed and crimes would be clearly and better understood. Hereditary diseases will be cured and study of cause, ill effects of various pollutants, radiations, mutations, etc. on human beings will be done. Now, what are disadvantages of Human Genomic Research Project? It may lead to some unethical approaches. Some countries can reprogram and reconstruct DNA and cause harm to other countries by infecting and then spreading it. Some countries can buy the rights of the project and control others. This is all about my today's lecture. Thank you.